Today, I will be explaining the synapses of chromosomes in meiosis given two individuals. Individual 1 has the following chromosomes, MLI.JKHG and GHI.JKLM. The dot in both represents the centromere. Chromosome GHI.JKLM can be flipped to be MLKJ.IHG, since the chromosome order still stays the same. When comparing the two chromosomes, we can see that there is a difference. The loci that are similar in both are M, L, G, and H. The genes where the chromosomes are different are I.JK. This is an example of paracentric inversion because the genes that were a part of the inversion also included the centromere. Now we will cross the two chromosomes to create the gametes and determine if they are viable. Since there is an inversion, the chromosome will accommodate for the inversion with a loop and a bubble. Here's chromosome 1. We have to add one more strand because each chromosome has a pair of two identical chromatids. Here's chromosome 2 and the second identical chromatid. Now we just have to add the genes on the chromosome and make sure the homologous regions line up. The centromere is located between I and J. However, in the problem we are told that there is a crossover event between the genes J and K. Now we must trace each chromatid to find the genes on each gamete. I'm going to use a different color for each chromatid so I don't get confused. The first one will be red for me and I'm going to trace along the first chromatid. The chromosome I end up with is MLI.JKHG. The second chromatid I trace will be pink. This one includes the crossover, so we will just follow along the loop until we reach the end. The chromosome I end up with is MLI.JKLM. The third chromosome is traced with orange, and I follow the loop all the way around. This one does not include the crossover and gives the chromosome MLKJ.IHG. The last one must be started on the right side because once a color is added to a chromatid, it can no longer be traced over. You can start on either the top or bottom strand, and I started off on the bottom. I followed along the line in the crossover event and ended up with the chromosome GHKJ.IHG. Now that all four of the chromosomes possible have been found, let's see if they are viable. The first and third ones are viable because it includes all 7 genes and a centromere. The second and fourth ones are not because they include duplications of a gene. The second one has 2 sets of the genes L and M, and the fourth one has 2 sets of the genes G and H. The expected fitness of individual 1 is 50%. Let's move on to individual 2. Individual 2 has the following chromosomes, MLI.JKHG and GHKJ.ILM. The dot in both represents the centromere. GHKJ.ILM can be flipped to be MLI.JKHG. When comparing the two chromosomes, there is no chromosome aberration present. No loop will be needed for this cross, and the chromosomes can be a simple line. 2 sister chromatids per strand and homologous regions need to line up. There is a crossover between the J and K region again. We will start tracing again, and I am showing the chromatid with the crossover event. Here, I finished tracing all 4 chromatids like individual 1. All the gametes are MLI.JKHG. All 7 genes and a centromere is present in all 4 chromatids, making them all viable gametes. The expected fitness of individual 2 is 100%. Here's a summary of both individual crosses and their expected fitnesses and my references.